It is a great pleasure for me to introduce our speaker today and to kick off what I think is a very exciting weekend full of light and illumination and hopefully bright insights. And before I do that, so I just want to make you aware if there's anyone who doesn't know about what's going to go on tomorrow publicly. Try the QR code and you will get to our webpage. I also would like to mention that this is possible through the generosity of a whole bunch of organizations. The fellows of the graduate students specifically have invited Dr. Nilsson, so he's our guest through the fellows of the graduate school. And then much of the rest of this working group is sponsored through the Office of Research that sponsors the Institute of Research and Science, of Sensing, which is doing a whole bunch of interesting stuff. Please check it out on the webpage too. College of Arts and Sciences, and also the Department of Biological Sciences. And before forward this one. Oh, because we need to go click back on this one. Yeah, there we go. Now, thank you. Before introducing Dr. Nilsson, I also want to make you aware that we have a little reception afterwards in a really special place that some of you may not know. It's the Chemistry Museum, and it has a lot of really nice older, museum stuff or museum equipment and really nice old books. So if you can make it, then make it there after this. It would be great if you could either follow someone or just so everyone knows we are now in old chemistry. So if you come back out and you have Simmer, the roof, there's the steps. If you go the steps down one floor, you'll be in the 5th floor, and then you go straight, and that way you enter in the 5th floor of River Hall, and then the museum will be right to your left. And if you get lost, look for 520. And with that, last but not least, let me introduce Dr. Dan Eric Nilsson. This is a huge pleasure for me because we have known each other for a while. Well, okay, first of all, here, as you can see, Dr. Nilsson has a whole series of awards, and I've been cheating by just putting them up instead of starting to reading them. Because instead, I think I much rather want to say a little bit how I got to know Dr. Nilsson, which is from the literature, back when I was in graduate school. And it seems like almost everything which I read at the time, had your name on there. And then I was kind of wondering, and eventually I got to go to this special meeting in Sweden. So Dr. Nilsson built the whole, so he got his PhD in Sweden, in London, then he went on to Canberra in Australia for his postdoctoral research, and then made it back to Sweden to just build this whole huge vision group, which now has become the epicenter of invertebrate vision. And as such, they're organizing this amazing meeting about every 5 years or so. It takes place in a beautiful royal castle near Lund. And it's very clever because they're kind of locking up, I don't know, close to 200 scientists, I guess 180 or so.
Yeah.
And so we're together, breakfast, lunch, and dinner, and get to play billiard in the royal habitat of some king from Sweden. And it's a lot of fun, and suddenly a lot of people that I got to know through this community and through papers then became real persons, and that's one of the most amazing things that happens when you study science. So it's a big group at Lund University now. Dr. Nilsson has done has published many papers in the book. There's a very important book, Animal Eyes. I consider it as our lab Bible. And is well known for his work on the evolution of all sorts of visual systems and how a complex organ such as the eye can evolve in the first place, which is one of the questions that Darwin asks. So he found a beautiful model for that. And I think without any further ado, please welcome Dr. Nilsson.
Thank you very much. So it's a pleasure to be here. But first, before I begin, I kind of would like to know a little bit about what kind of audience I've got. You're not all visual scientists, are you? No. Because I can't change my slides, but I can change the way I sort of explain things quite a lot for basically any kind of audience. So I should try to take it on a much broader audience. Is that right? Yeah, then we'll do it that way. We'll do it that way. So the idea was to say something about the evolution of, actually, I should change the slide, the evolution of eyes and vision. And in particular, I would like to bring some more clarity into what was driving it. Not so much looking at genes and genetics and things like that, but what was actually the things that was, or the things that were the selection that was driving evolution of eyes and vision. When I started to investigate this, I kind of just thought about eyes, but then as I went on over the decades, I realized that actually I'm not really investigating the evolution of eyes at all. I'm investigating the evolution of vision, because that was what driving it. The eyes are just things that have to modify and evolve in order to provide vision for the animal. So it looks like now I'm going to talk about the human eye. I'm not. Well, partly I am, because the human eye is an animal eye, because humans are animals, right? We're going to talk about all kinds of animals and all kinds of eyes. You're kind of familiar, I guess, with the eyes that animals have on their head, pairs of eyes that animals have on their head. You find that in all other vertebrates. You find it in birds, in lizards, in fish, and so on. You also find it in, know what that is? It's actually a squid eye. Squid have beautiful eyes too, pairs of them. That's a little spider. It's got paired eyes, got more than one pair. That's a compound eye of a horsefly, and that's a compound eye of a trilobite. They're not around anymore. But anyway, there are lots of these large, nice eyes, paired eyes on the head of animals. But there are also animals that have much less obvious eyes, like little velvet worms you have there, snails, ragworms, and some flatworms. They all have paired eyes on their head. And you can't dismiss those, because actually by studying those, you start to understand how those nicer, bigger things evolved. There's no way you can understand eye evolution by just looking at the end product. No way, no way. You have to look at the simpler stages as well. And then other eyes as well. There are eyes which are not paired, eyes that are in the middle of the head. Many animal groups have got those. Some lizards have them. Insects have little triplet of eyes there. Some flatworms have compound eyes there, and some crustaceans have little funny eyes in the middle. They're all right in the middle. Some have that just as the only eye they've got, and some has it together with paired eyes. Then we've got eyes that are not actually on the head at all. Eyes that are distributed in various parts of the animal. Like on clams, you've got eyes along the edge like that. Some of them have little funny things that look like blueberries. They actually use curved mirrors for forming an image. Others have compound eyes. It's not a scallop, it's another clam that has that. You find peculiar eyes on fanworms. That's fanworms, a kind of polychaete worm. Well, it doesn't tell you anything. You don't know what polychaete is, but it's a marine worm you find in the sea. Some of them have reduced their head and formed a little tentacle crown, and some of the tentacles bear a new eye. They've actually reduced their normal eyes on the head. They're not entirely gone, but they're nearly entirely gone. You can find some small rudiments of it, and they made new eyes. Starfish have little eyes at the end of their arms. That's a box jellyfish. They've got beautiful eyes. So there are lots of funny eyes that are not actually on the head. And on top of that, you've got animals that have vision. They have a visually guided behavior, but they've got no eyes at all. Is that possible? It is, because you can actually have distributed photoreceptors. You have light-sensitive cells all over the body. This sea urchin has cells that are over in that side. Of course, they don't look in the other direction. They point in that direction. So the whole animal kind of becomes a big compound eye. You find that in a large number of different animals. So across the animal kingdom, there is an absolutely astonishing diversity of eyes and light-sensitive cells. And that is what we're going to try to understand, how that evolved. Then we must, a way to kind of tackle this problem is to ask, what do all these things have in common? Is there anything they have in common? Maybe lenses? No. Many of them don't have any lenses at all. You can look at all sorts of structures you have in your eye. You will fail. No, they appear in a few cases, but not in all. There is one thing they do have in common, though, and that is the light-sensitive protein that they use to detect light with. They're called opsins. These little proteins, they are related to other receptor proteins that we have in that all animals have in their cell membranes. They do all sorts of things to transmit signals from one neuron to another, or they are involved in sensing hormones, and they are involved in smell, for example, olfaction. A particular type of them are called opsins, and these proteins themselves are not light sensitive at all, but they bind a little vitamin A molecule, and it's the vitamin A molecule that can pick up light energy. When it does, it flips to another conformational state. You can actually shoot it back to that state with another wavelength of light, or you can have an enzyme that folds it back. So the good thing with this molecule is that it can be recycled. You use it over and over and over again, the same molecule. You don't consume it. So then you might wonder, where did these opsins come from? Because it turns out animals have them. Nearly all animals. Not quite, though. There is one little group of animals that do not have them, and that is the sponges. They don't have them at all, but all other animals have them. Sponges, though, they have some, or actually, if we look at these opsin proteins and ask what is the other receptor molecule, because that's a huge family of these receptor molecules, what is the receptor molecule that is most similar to opsins, which is most closely related? That turns out to be a melatonin receptor. Melatonin receptors look almost identical, but they don't bind these vitamin A molecules. Instead, they bind melatonin. And it turns out that the melatonin molecule is actually itself light sensitive. If you shine light on it, it oxidizes and can't be used anymore. So you have to produce new melatonin molecules all the time. And if you look at sponges and sister groups of animals, they are little colonial, tiny few cell things. It turns that they can't produce any opsins, but they produce melatonin receptors. They have enzymes that can produce melatonin, and it's quite likely that they have a constant production of melatonin. They are small, transparent things. At night, that builds up to high levels. They get high levels of melatonin, which means that their melatonin receptors will signal that there is lots of melatonin around. At daytime, when there is light, all these melatonin molecules will be oxidized and removed. As soon as they are produced, they will be removed by light, which means that melatonin levels are low. So these proteins will not sense any melatonin, and that will tell the little cells that it is most likely daytime. We have preserved that role. I mean, melatonin is now, for us, a hormone that tells us that it's night. That's exactly what it did before too, but it was also a light receptor before. Now it has become more specialized. And vitamin A is a way better molecule to pick up light than melatonin. That one is removed. You have to produce new molecules all the time. These ones are recycled. That is sensitive in the blue and UV, and it's very hard to change that sensitivity. You can change the spectral sensitivity, the wavelengths it's sensitive to with vitamin A very much by just basically when the molecule comes in here, you can put it in some strain, bend it a little bit, and these bonds become more slack and they pick up longer wavelengths, or you can make those bonds more tighter and they will pick up shorter wavelengths. And you can do that over any way from ultraviolet up to red. So it's an amazing molecule, the vitamin A molecule. Now it turns out that if we look at different groups of animals, like what is called deuterostomes, that's where we belong, all the vertebrates, Lophotrochozoans, there you'll find octopus and squid and snails and things. There are also flatworms and all sorts of soft, gooey things, ecdyes and so on, slightly harder things like insects and nematodes and various other things. Cnidarians, that's jellyfish and corals and things. All these, they actually have a fair number of, a small number of different types of opsins. But they all have the same types, which means that when opsins first evolved from melatonin receptors, they seem to have diverged into a handful of different classes. So that happened really early because they all share those classes. So it seems like this is kind of a phylogenetic tree of opsin evolution from the melatonin receptors. You find that in sponges, but you find melatonin receptors in all these things too. So the melatonin receptors didn't disappear, they're still used. But opsins appeared and they immediately diverged into several different classes or families, which are now found in all animals. But if we try to look at this on a timed evolutionary or timed phylogenetics tree of all animals, so now it's not opsins or anything like that, it's now animals. So we've got the different animal classes, we've got the sponges down there, we've got the jellyfish there, and we've got the other three classes of bilaterally symmetric animals. And now this is a timed tree, so you've got millions of years on this scale. You can see when they actually divided and branched. If you talk to people that are in this field, they will be really mad when I say that those things branched at 800. One person will not be very mad, but all the others will be mad because they say, no, it's there. And they don't agree at all. I don't care so much, actually, where that happened, but it's right. I think I kind of picked the one where you got the highest numbers here, but you could also get possibly 700 million years or so. But anyway, it seems like roughly around 800 million years the opsin molecules appeared. Put that in there. So opsin molecules appeared 800 million years ago. And when had they diverged into the major classes of opsin molecules? Nearly immediately after that. So then the light sensitivity that we find in all animals except sponges was established roughly 800 million years ago. That's a long time ago. So what was it? If you have just an opsin in a cell, what do you get then? What can you use it for? Because that's really what we want to look at now and see how can this benefit animals in any way at all? If you have a light sensitive cell, it will tell animals about the time of day, which is really, really important, especially for small animals that live in the sea. They need to know if they swim, if they are planktonic, they need to be able to regulate their depth, to be at the right depth, not too high up in the day because there's too much ultraviolet, but fairly high up because they actually eat other algae and things, single cell algae and so on. So they need to be at the right height. And at night, when those things are not available, they have to go down to protect themselves. So they need to migrate up and down. They need to know what time of day it is. And they also need to be able to control their depth. So they can actually use their light sensitivity, which is then a non-directional light sensitivity, because you can imagine these are just animals with very few cells. Transparent animals that didn't have any dark pigment. So these light receptive cells, they picked up light from all directions. This is non-directional photoreception, which can then be used apart from checking the time, also determining how deep the animals are, possibly as shadow detectors, although that may not have been the first things to evolve. But you can kind of see that if you want to determine how fast changes are, because that's a good way to discriminating between depth and time of day, then you need two different types of photoreceptors, one that is fast and one that is slow, and you compare the two, and then you get a time signal. Or you can see how fast things are changing. Or you can have two different photoreceptors, one that is sensitive in green and the other one that is sensitive in blue or ultraviolet. And then you can also discriminate between what is time and what is depth. So you need a number of different types of photoreceptors with different types of opsins in, but we saw that they diverged really early, and this is possibly the reason. You need to have a number of different classes of them with slightly different properties to get this information out. Now we're going to try to put this into a kind of an evolutionary scheme. So we start with we just assume that everything started with very simple, unspecialized, light-sensitive cells, and that led gradually to more and more advanced eyes. We kind of assume that. We'll see that it really makes sense when we put in the other things, but that's the kind of structures, because we see all these things in animals today. They are all existing in different types of animals. But these are structures, and what we really want to do is to look at function, the evolution of vision rather than the evolution of eyes. So we're going to make a little schedule scheme here and see how vision evolved. And we can already put in the first step, namely the non-directional photoreception, which we've already talked about. And that is then used for light cycle tracking, as we said, and also for some other responses like depth control, shadow detection, and exposure detection for animals that actually live in the bottom, that bury in the seafloor. They need to know when they actually get through the surface, through the bottom, and get exposed to light. So, and animals that do just this, there are many today. This is a sea urchin larva. It has non-directional photoreceptors. That's the only kind of light sensitivity it has. It cannot determine where light comes from. It is just a non-directional photoreceptor. So you see no dark pigment. It contains no dark pigment. But there are actually several types. There are like 5 different types of photoreceptors in them. If we now want to get more use out of this photoreceptor, what kind of simple thing can we possibly add? Screening pigment. Some dark pigment that makes sure that light can reach this cell only from a restricted part of the world, not from the rest, which means that if the little animal, because they're still tiny, moves around a bit, it will make sure it will sense that it's brighter over there and dimmer over there. If it wants to move towards the brighter areas, if that's good for the animal, it can do that. This will guide the animals. It's called phototaxis, and it is a really common ability that many animals have, especially really small things. They can't see an image at all. They can just move towards light or away from light. Hi, Todd. So now we've got another level already established, which is then directional photoreception, which is good for orientation, which is it's a new and really powerful thing. If you can orient in your environment and find the best parts in the environment, really important for animals. But before we continue now, we have to talk a little bit about problems with detecting light. As soon as you detect light, you want to know how much light there is right now and a second later, or how much light there is in that direction or that direction. You have to compare measurements of light. In principle, you have to count photons, and you have to determine how many photons do you have in each sample. And then it turns out you can't have too few photons. If you have very few photons, like in the blue ring up there, you see there are 3, 4, 3, and lots, and that varies because photons arrive randomly. And you need to, if you want to see differences that are about 30% differences, then you can do with about 10 photons in each sample, roughly. If you want to be more accurate and see 10% differences, then you need about 100 photons. If you want to see 3% differences, you need about 1,000 photons in each sample, and if you need to see 1% differences, then you need 10,000 photons. As soon as you want to make more accurate measurements, you need way more photons. And the problem is now what we did, we started with a thing that measured light from all over the place to light that just came from less than half the world. And if you do that, you can't measure light over a long time either, because when the time changes over the day, you can measure over 10 minutes and then have another sampling period for 10 minutes. When you want to use light for orientation, you have to have much shorter shutter times or integration times, which means you get very many fewer photons in your samples. So the consequence is you will suffer from photon shortage, and that's bad. It means you can't use your system. It doesn't work at all. And one of the main reasons you would, and you can make calculations, it turns out the non-directional photoreceptors will work fine. When you start to make them directional, they won't work any longer. simply because an unspecialized cell with some opsin molecules, as many as you can pack in the membrane, you can't pack too many because it will interfere with normal other proteins that are in the membrane that serves the normal metabolism of the cell. So if you have too much opsin in there, it'll actually be toxic. So you can't have too much. And the amount you can have means that light that goes through this cell, you can pick up about or less than 0.005% of it. Nearly all the light just goes through. It's not very efficient. It's basically a hopeless photoreceptor. But for tracking, seeing the daily changes of light and for measuring depth in the sea, it works because you can be slow and you pick up light from all over the place. It does not work for directional photoreceptors. So we need to do something about it. And the one obvious thing to do is, of course, to specialize the membrane. So you have a specialized part of the membrane where all the opsin molecules are. Then you can free the rest of the membrane to do the normal things to keep the cell alive. Once you've got this specialized region of the photoreceptor membrane, then you can start to fold that and stack it, make larger and larger stacks. And then you can easily absorb more than 50% of the light compared to 0.005%, which is a huge gain in sensitivity. And then suddenly phototaxis works easily, no problem. And photoreceptor cells that do these things, they have these stacked membranes. This is what it looks like in our rods. That's actually a rod, but our cones look fairly similar. We have discs or lamellae of flat membranes lying stacked on top of each other. In insects, for example, and most other invertebrates, we find microvilli, little finger-like projections instead. Another way of just producing very large amounts of membrane in a small space. This is what these directional photoreceptors typically look like. There is a pigment cell and there is a sensory cell, and the sensory cell, photoreceptor cell, has some of these membrane specializations to put their opsin in. Animals that have these are, here are some examples. Flatworm, and that's a larva of another. There's a rotifer, and that's, what are they called? It's a tardigrade, yeah. It's a bit hard to see actually here, but there are dark eyes there. It's a dark thing there, a pair of dark things there. Each of these things it's not an eye. They do not see an image. They just measure light in a direction. They're used for phototaxis. So can we now possibly improve this and make it even better? Obviously, yes, we can. Rather than just having one photoreceptor that measures light in a broad direction like that, we can have more of them pointing in different directions so we get an image. Then we get a real eye. So let's do that. We've got them up here. There are many different ways of doing it. You can put all of the photoreceptors in one pigment cup. Then you start to develop a single aperture eye, which eventually ends up with the type of eyes we've got. You can do it the opposite way, so you have eyes or little cups pointing in different directions out from the center. Then you start to develop a compound eye. Or like some animals, you can actually have them dispersed. Like directional photoreceptors with rather narrow angles spread all over the body. Some animals do that, but of course, these two solutions are by far the most common, actually make a real consolidated eye. And then we can add that to our general scheme, and now we've got the third step, which I have called ancient vision, but the reason I've called it ancient vision is that what I really would like to call it is non-object-based vision. But that's kind of square in the mouth, so I've shortened it to ancient vision because it also is the first type of real vision. So let's call it ancient vision, actually means then non-object-based vision. Animals use that for determining their self-motion. They see the optic flow as they move. They can determine the distance between themselves and various structures around them. They can find free paths to move so that they don't swim or crawl into obstacles. It will help animals move around in their environment, and animals can find their habitats, find the places to be much better with this ancient vision, which is then based on what we could call low resolution eyes. They can do that far better than they can do with a directional photoreceptor, which means that when this evolves, you can skip that, drop that. So that turns into that, and that disappears. But now we've done something again. We've reduced our angles again, and with small angles, motion, the time for each sample that you measure photons in have to shrink even more, otherwise there will be motion blur. So we end up with, again, photon shortage again. But now we've kind of used the strategy of fixing the membrane things, so that idea is gone. It's already been used. Is there any other way? Well, there is one of the problems is actually that the photoreceptor cells, they are really tiny. They're like 5 to 10 micrometers at the most across. That's a really tiny thing, and that doesn't pick up much light from one particular direction in space. Easily fixed. Put in a lens. And it is the diameter of the lens that determines how much light you get from that point in space. And the lens, of course, can be much bigger than the photoreceptor cell. So this turns out that this solves the problem again. And low resolution eyes, ancient vision works if you put in lenses. You can actually do it without lenses if you're all right with a really crude image with very few pixels. But if you want to just increase the pixel number a little bit, you need to put lenses in there. The reason to put in lenses may not actually originally have been to increase sensitivity. There are other reasons too. UV protection, put in something that takes away ultraviolet light because it damages the photoreceptors. It could also be mechanical protection. Imagine you've got an eye like that. You don't want little creatures to crawl in there and think it's a cozy place to be. So put in some mechanical protection, then you can possibly make that mechanical protection also absorb ultraviolet light. But then you've got exactly what you want when you need to create a lens. You've got a transparent material there, which you can increase the refractive index of, and suddenly you've got a lens. Animals that have these low resolution eyes used for ancient vision, here are some examples. That's a flatworm. They don't have any lenses, so they use very low resolution. That's a ragworm. It's got 2 pairs of those things. They contain something lens-like inside. That's a box jellyfish. It's got lots of these little eyes in funny structures around here, and they contain a lens. None of these eyes are particularly well focused. It doesn't matter. They're not really out for high spatial resolution. We can take a little look at that. This is a velvet worm. They have these tiny eyes. They're used for orientation only. They're not actually good enough. They don't see each other. They are predators, but they don't see the crickets and things that they actually eat because they move up really close to them and sense them by smell and other senses, and then they spray glue on their prey, but they don't actually see their prey. They use their eyes to be able to find shelter because they're night active. At dawn, they need to find shelter because otherwise they'll dry out. In the evening, they have to find a way out of the shelter, out in the open where they go hunting. This is what we would see from their natural habitat. That is what they would see. And to us, it looks like, oh, what a crappy image. That's absolutely useless. And if you go to an ophthalmologist and say that your vision is like that, he will call you blind, basically. But these animals aren't blind. That is good enough. That's perfectly good for these animals. They can do orientation. They can orient to that. They find the right habitat. They know to be in the right place at the right time with that kind of resolution. This is what a box jellyfish would see if it went on a roller coaster ride. They may not actually do that very often, but I know you've always wondered what the world looks like to a box jellyfish when it goes on a roller coaster ride, and that's what you see up there. Fast things are gone, fine structures, fine details are gone, but that actually helps the box jellyfish to stay in exactly the right height. This particular species that we have up there live in mangrove swamps, in particular parts of the mangrove swamp. You can use those eyes to put it in exactly those places. They don't see each other, they don't see their prey, but these eyes keep the animals in the right place. Okay. So can we improve these things any further? We've actually got one layer left on our diagram, so we need to do something. Yes, we can make higher resolution eyes. Why would you make higher resolution eyes? We've just concluded that it's fine for these animals to use these low resolution eyes for ancient vision. But you use your eyes for more than that. You actually use your eyes for ancient vision a lot. When you walk around it, you use them for exactly the same thing as fox jellyfish and velvet worms use their eyes for. But you know that this is a slide switcher and a pointer. So this is an object. I am an object. And you're all objects. Your pets are objects. A fly is an object. We divide our world into objects. And animals can actually not necessarily have as many different objects as we have, but many animals are able to discriminate between objects. But that is, and then you need to basically do the same things as for ancient vision first, the same kind of processing stages. But then you also need to add segmentation of the object to actually see the object against the background. You can use motion cues, color cues, intensity cues, pattern cues, all sorts of things to make sure that that is an object and it's separated from the background. There's another object there, so you need to discriminate object because you can't have the same behavior to all objects, and it would be pointless. So you need to be able to classify at least a few objects. It's that type of object, like a prey animal, and another type of object, like my own species. Possibly the opposite sex or something like that. So you know those two different objects, but then you need to have certain behaviors in response to some objects and a completely different set of behaviors to another. And that's really powerful with object vision. You can make so much more advanced behaviors with that. But if you want to be able to discriminate objects against the background and see many objects in your environment, then low resolution is no good. You need high resolution. You need much higher resolution. And if you need higher resolution with really narrow angles for each photoreceptor, narrow angles again means you need to speed it up, because otherwise you'll have motion blur again. So of course, we'll soon get to that, but we can just first add the last stage, object vision for interaction with objects. Really powerful, fantastic invention. But since it requires many more photons, because we're also going to see, it's also very small differences in intensity we need to see in order to make this work. And we get very few photons. So again, photon shortage hit again for the third time. And it seems we've already used all the tricks there are. Is there any possible way we can fix it this time? There is, luckily. The previous animals had small eyes. Just make them large and you make this area large again. You need to make really large apertures. But if you have large apertures, that means large eyes. No, well, not this large, this large. Instead of a tenth of a millimeter, you need eyes that is a millimeter or more, something like that. But up to centimeters or a giant squid of eyes like that. So you need to make a much larger eye. And in order for animals to carry a larger eye, they need a much larger body. In order to act on these things, they need muscles and things attaching to skeletons. All sorts of new things became needed. And we can just take a short look at which animals this actually concerns. But of course, then it's actually just a rather small part of the animal kingdom that has done that. It's the vertebrates, where we belong. Well, we've got vertebrates there, yes. Cephalopods, that's octopus and squid and cuttlefish. They've done it. And arthropods, spiders, insects, crustaceans. They've done it. So 3 major animal groups have gone from ancient vision to object vision. And they have become large. And they have also all developed large brains, because this takes a heck of a lot of neural processing to do that. So you really need a large brain. When did it happen? When did large animals appear? In the Cambrian explosion 540 million years ago. So may actually have been so that the evolution of object vision was the driving force that pushed the Cambrian explosion. And you can imagine a single species starting to become a visually guided predator. That would put an enormous pressure on the rest of the ecological community. They would all have to be able to see that nasty bugger that was fast and ate them in order to escape. And when the animals start to see a lot themselves, they can also, some of them can start to become visually guided predators themselves and start to see each other and communicate with vision. And there's so many things you suddenly can do. But then you need to be a lot larger. So that was what happened during the Cambrian explosion. If we look on this time scale we had before, we saw that the first opsins evolved here. The Cambrian explosion, 540 million years is there. Sadly, for all that time after that, nothing really much has happened in terms of eye evolution and vision evolution. It all happened there, was done, ready. I mean, eyes and brains and vision has evolved and changed tremendously since then, but not principally. The major tasks or types of vision and eyes were already in place 540 million, or roughly there. It depends on where you think, if it was, the Cambrian explosion didn't go in a second. It was, after all, over a time scale that would appear long to us now, but in a geological perspective, it's short. So now we can start to ask, how many times did this happen? It was once. Each of these steps, did it just happen once? The fact that we got camera eyes and the compound eyes kind of indicate, no, it must have happened more than once. But let's take a look at the different steps. The first step to opsin, that happened once, only once. The next step to directional photoreception happened a lot more often, at least 13 different times. I think Todd over there will possibly suggest even larger numbers, but I say at least 13. There are certain things you can be certain about, different things, but there are some we don't know yet. But we'll find out more support behind that and actually crank that number up, I think. If we look at lenses, at least 4 times to compound eyes and at least 11 times to single aperture eyes. If we go the last step, large size, at least once for compound eyes and at least 3 times for camera type eyes. So these things, it means that since these things seem to occur over and over again, then the principles of evolution of eyes and vision is not sort of something, it really is a principle. It happens the same, it follows the same path every time for every group. That is the way it has to happen. And the reason for that is, of course, physical limitations. You need membrane stacking, you need lenses, you need large size. There is no way to escape that. The physics put boundaries on what you can do. Right. So now we can ask, is this all there is to the evolution of eyes and vision? Have we reached the end? Is this the end of the talk? That's not. There are more things. Let's say now, we've already worked out that there is ancient vision and object vision. So we can divide vision into major domains, which are rather different in what animals can use them for. And we know that the first animals had just ancient vision. We have both. Because when object vision evolves, it doesn't mean that ancient vision in any way becomes superfluous. It's just as important as it ever was before. But the eyes are better, because you need better eyes there, so that the rest you can actually do those things a bit better as well. So that's one way of dividing vision. But we can divide vision in a completely different way, namely in action vision, which is kind of see and act. When animals or humans move and do something which is visually guided, feedback comes in through the eye and corrects the movements and makes sure we reach our goal, whatever we want to do with the behavior that is going on. Anything that has happened just a fraction of a second ago is of no interest at all to action vision. It doesn't need any memory. It just works forward towards a goal. And then the other thing is perception, which is kind of the opposite. To pick up information that is not to be used immediately. It could be used a couple of seconds later. It could be used minutes later, hours later, days or years later for various reasons. But that is to pick up information about, to assess the environment. And that's what psychologists call perception, I think. So it has a possible influence on behavior, and from very short to very long time constant for that. If we combine those two, that means we will actually divide vision into 4 quadrants, or 4 major domains. So we have the combination of ancient vision and action vision. That's visually guided orientation, right? Lots of neurobiologists study that. If you combine object vision with action vision, then you get visually guided interaction with objects. Lots of neurobiologists study that as well. It's well-known things. If you look at object vision and assessment vision or perception, then that's object perception. Lots of visual psychologists study that. And then you've got the combination of ancient vision that is non-object based and assessment vision or perception. Then you get into something that, hey, no one really works on that at all. That's an unexplored part, but a really important part, because animals can actually use that. And before object vision evolved, in all those groups, they only have got that, but they cannot only do with that. They also need to know where to go. It's not just to do something. They need to work out that I want to go in that direction because that looks better. That's where I want to be. And not just that, there is something that is actually even more important. Animals have a behavioral repertoire. More advanced animals can do more different behaviors, but all animals can do a fair number of different behaviors, and they should not be picked randomly, because that's suboptimal. Under certain conditions, certain times of day, certain weather conditions, certain environments, you should do some behaviors. Under other conditions, you should do other behaviors too. So this master control of behavior is really important. And when you only have this part of vision for all those animals, that is the way they can do that. Of course, there is lots of other information, other senses as well, that goes into what I call, what people in general now call behavioral states. For humans, you would call it possibly something else, mental states or so. But behavioral states are set by all sorts of external information. Vision is really important there. And there are also internal information about fatigue, thirst, hunger, sexual state, all sorts of other things that tells the animal from the internal senses which contributes to setting a behavioral state. But once the behavioral state is set, it means that the animals have an interest in doing certain things, no interest at all in doing other things. And this part is really important for that. And that is actually now we can add to our general scheme that for each of these levels we have action vision and we have ancient visual perception there, and we have object perception up there. So it now becomes a little bit more complex, but more complete. Interestingly enough, this unexplored part actually has much to do with light cycle tracking, the biological clock and all those things. They're kind of related. They serve roughly similar things. So these, if we look at nervous systems, we'll probably see that these things are processed in similar places. It could also be actually that the evolution of these things did come straight from here. So maybe we have to add some extra little lines here in this diagram. Now let's end this talk by shortly looking through the kind of information that animals use to do these things. For orientation, for ancient vision in combination with action vision, then that's contrasting borders. When you see an edge of something, there is bright there and it's darker there. Those are the things that are coded, and their motion. So the optic flow, that gives all that information that animals need for that. For interaction with objects, then of course objects have to be segmented from the background, and particularly and they have to be recognized so you know which type of objects there are. But for action vision, for object vision and action vision in combination, interaction with objects, you also need attention, because you can't attend to all these objects at the same time. So you need mechanisms that focus your attention to particular objects. For object perception, on the other hand, you probably should have a better idea of many of the objects and what they are doing. because that gives you an idea of the situation, what is actually going on right at this moment, and things that you can use for determining exactly what thing you're going to do. But these behavioral states that are set then by the general background, and that is a bit tricky since this is something that is really unexplored. We don't know precisely what kind of information there is, but we can maybe find that out. So if we try to look at what you can actually do with this fourth quadrant of vision, what kind of information you can get from it, it is like searching for good habitats to work out a good place to be. Search to decide about if you want, what kind of behaviors you want to do from the behavioral repertoire, like searching for food, pursue prey, protect against threats and things, interactions with other animals of the same species, grooming, cleaning, caring for nests and burrows and shelters, and resting and sleeping, which are also behaviors. So you have to decide about what you're going to do from this long list of which can be even much longer, of course. And if you look at these two places, if you imagine you were there or you were there, you would probably feel like doing slightly different things. But we are humans. We are extreme generalists, so we would be happy in any of those environments. We would find something to do. For most animals are not as much generalists as we are, nor do they have so many different objects and things to think of. So they have to rely much more on these cues. So how can we extract reasonable cues from this? How can we work out that in that environment you should do certain things, and in that environment you should do other things? How can that be worked out? If you look at these are two different scenes from the same environment. These are actually photographs that cover 180 degrees. So that's straight down, and up top there is straight up, and then it's 180 degrees in the other direction as well. These are two scenes. They are taken about 10 meters apart and in different directions. So scene-specific information is kind of useless here. But if you have a really slow visual system, or a slow part of your visual system, you can start to integrate over many scenes. Now you have 10 scenes there, for example. If you integrate over super many scenes, you get a vertical light gradient. You can divide that vertical light gradient into different components. For example, if you have, so this is minus 90 degrees, plus 90 degrees, that's straight down, straight up, that's the horizontal plane. If you look at the intensity here, you can see that the intensity on a log scale here, so that's 10 times brighter than that, changes like that in this particular environment. The range of different intensities, which is a very good measure of how much visual structure there is, changes in this way. Or in the dark gray, you've got 50% of all intensities, and the light gray, you've got 95%. So that tells you that there is much more visible structure here. Everything becomes much more homogeneous when you look up in the sky, right? So that's one type of vertical light gradient. You can also look at different colors. Here I've just compared blue, green, and red, and they behave in a very particular way in this environment. So and you can also look at the dynamics, which I haven't done here, but you can do that and see how much things change in different speeds and in different parts of, or in different elevation angles. If you make such measurements in different forests, you'll find out you can actually see a rather distinct difference between different forests. If you look at semi-open environments, oh yeah, they are very different. If you look at really open environments like deserts and meadows, oh yes, they are pretty different. If you look at underwater environments, they are pretty different too. If you look at the same environment under different weather conditions, oh yes, there are very specific things which happens with these curves. And you don't need to have these high resolution. You need a few points here and a few points there to be able to determine what kind of environment, what kind of weather it is. You can also determine time of day, because these curves shift up and down on a log scale. And here you see different colors. Look, they behave in different ways at different solar elevations, even if the sun is above horizon or below horizon, you can kind of read that. So that's a pretty good timing device. You can also work out seasons, drought conditions. You can work out all sorts of things. So type of environment, weather conditions, time of day, season, depth in water if you're aquatic, you can also include there. So which eyes do this? In insects, I first thought that maybe the normal eyes are possibly used for that, but it turns out that if you paint over these, which are called dorsal ocelli, the median eyes, insects become really useless in picking the right behavior. So it seems that insects really use the median eyes there. What about us then, vertebrates? We have the retina, our normal eyes, but we also have, well, actually we don't, but mammals don't have a pineal. We have a pineal organ, but it's not light sensitive. In all other vertebrates, in fish, birds and lizards, it's light sensitive. And there is also another organ called the parapineal, which is a median cluster of photoreceptors. And some lizards have these parietal eyes, which are also part of this system here. If we take a look at, so we've got these median photoreceptor structures, which sometimes are actually median eyes, and we've got the real eyes. If we take a look at the neural circuitry of these things, we'll find out that in the retina, we've got photoreceptors that are based on cilia and has got the class of ciliary opsin molecules in them. They are connected via a bipolar cell to neurons that project into the brain. They're called retinal ganglion cells. Some of those are light sensitive, and they contain a rhabdomeric, a type of opsin that goes into those finger-like projections that are typical for rhabdomeric photoreceptors. If we look at the pineal, it's exactly the same thing. The bipolar cell is missing, but we've got a C-opsin photoreceptor connecting to an R-opsin photoreceptor. Two different photoreceptors. The similarity between our median photoreceptors and our eyes is just striking. It's unreal. Is that right? If we look at other groups of animals, it turns out they've got rhabdomeric receptors, generally, most of them, in their lateral eyes. In the median eyes, if they have median eyes, so they usually have median photoreceptors at least, they are most often ciliary receptors. Sometimes they are rhabdomeric, and in a few cases, you've got both. That's what we look like. We've got a combination of ciliary and rhabdomeric in both the median eyes and in the lateral eyes. That kind of suggests that our lateral eyes are not really lateral eyes at all. We may actually have lost our original lateral eyes long back in the Cambrian explosion, and then formed new ones. So if the median eyes formed lateral cups for reading the vertical light gradient, then that may have turned into new eyes, new lateral eyes. That actually explains exactly why we have lateral eyes that look completely different in terms of their neural structure and molecular signature to the lateral eyes of all other animals. So, but that sounds kind of weird. So we don't actually have lateral eyes. We just have median eyes that have turned into lateral eyes. Is that believable? Has it happened anywhere else in the animal kingdom? Oh, yes, it has. Copepods is a small group of crustaceans. They are tiny and they have lost their lateral eyes, except some species have actually made new lateral eyes out of their median eyes. Jumping spiders, they have beautiful lateral eyes, appearing to be lateral eyes, but they actually date back to the median eyes of their sea scorpion ancestors. So it has happened before, which means that these glasses may not actually look as ridiculous as they seem. Thank you. Right, questions?
Great. So you would probably take our eyes as like your highest classified D category, right?
Yeah, definitely.
But what about our peripheral vision? Because
peripheral vision is largely concerned with orientation, so that's largely ancient vision.
Okay, that was what I was thinking.
Yeah.
And
we don't really do much object discrimination in the periphery, but when we move, that's what tells us where we don't walk into things, and we know how fast we move. We know if we walk straight or if we bend. All those typical things that
we don't normally pay that much attention to it.
No, yes, but it is really important for our behavior. Yes, yeah. We can't just rely on objects. Then we would fail totally. We need the optic flow. And that is what peripheral vision largely does. Yep.
Thank you for your talk. I think my question was geared towards the slide. I'm not an expert in what you're doing, but I think it was geared towards the slide where you had your membrane with the oxygens like surrounding it, and I think it was titled photo storage or photo shortage, and I don't remember.
Yeah, photon shortage. Yes.
Shortage.
Yes.
Okay, I thought it was storage because like
No.
like you had the oxygens around it, and then like you started stacking them. I think.
Not oxygen. No.
Okay, can you explain that? Because I think I completely missed it.
Yeah. Let's go back. Let's go back to that. Yeah.
I thought it was cool. I saw the efficiency went up, so I didn't know. Yeah, it looked like
Go a long way.
The beginning.
Yeah, that was close to the beginning. Yes, it was. No.
You're thinking of the opsin genes, not oxygen, opsin.
Oh, okay.
Yeah, the gene, so it's capturing more photons.
It was this slide, okay.
Yeah.
I thought you said opsin, but you said apsogene?
Opsin.
Opsin, that's the protein that is keeping the vitamin A molecule, light-sensitive vitamin A molecule. So that's another name for it is the visual pigment.
Okay.
So the visual pigment is the opsin molecule plus a vitamin A molecule. That forms a functional visual pigment. So, yeah.
So then, like, what's happening in this, like, when you were stacking them? Like,
when we were stacking them, it's a good way of absorbing more light because there was a photon. These things didn't absorb enough photons to be able to discriminate different intensities. You need to absorb more photons, more light. And then the way of doing that is to make a special part of the membrane where you can pack opsin molecules much more closely. Because then you can free the rest of the membrane because it needs to do other things as well to keep the cell alive.
Okay.
And once you've made a special region like that, you can stack them and make many layers, which means that each layer possibly absorbs like a few % of the light. But then add layer after layer after layer, and you come up to 100% if you make this stack long enough. Well, almost 100% at least. In reality, it turns out that animals pick up day-active animals, which have sufficient space to make long photoreceptors. They pick up somewhere between 50 and 80% of the light. Some deep-sea animals can go up to 90-95%. Night-active animals, they pick up remarkably a little bit less. But yeah.
Are there any animals that have eyes that can perceive UV light?
Oh yeah, many. We belong to the few poor suckers that don't. Birds.
How do they do that if it's destructive to the photoreceptors?
They just have to. I mean, blue light is destructive too, but not as bad as ultraviolet. So we need mechanisms to fix up because it forms free radicals which are aggressive. We need mechanisms to remove those from our photoreceptors. If you also have UV light hitting them, you need to have those mechanisms have to be much more active, and that's costly. Plus, sometimes it goes wrong anyway. So there is a price to pay for it, but there is also a gain in it because there is information in the ultraviolet. We don't use it, but mice, for example, they're dichromats, so they only have green sensitive cells and ultraviolet sensitive cells. We have blue, green, red cells, but mice have their dichromats, but they have got cells sensitive in the ultraviolet. Birds in general, they are tetrachromats. They've got red, green, blue, and ultraviolet. Some birds have violet, others have ultraviolet. Fish, generally the same thing as well. 4 different types, and they nearly always go into the ultraviolet a bit. Insects go even further into the ultraviolet, really deep in the ultraviolet, but not so far up in the red, unless they are butterflies. They do it. Most insects, like bees and so, they have ultraviolet, blue, and green photoreceptors. And ultraviolet is an important spectral range for them. But they've also got smaller eyes. Light passes a shorter distance, which means it's easier to make the ocular media, the lenses and things, transparent to it. It's really hard to get UV to go through a large eye because ultraviolet does absorb in protein in general. So for animals that have slightly smaller eyes, it's easier to do it. And there is information in the world that is useful. Yeah.
I have a naive question, I'm sure. You showed that there were actually members of attempts to, as you go up the evolutionary ladder, but only one attempt at the opsin molecule level.
Yeah.
But can one imagine, from your knowledge of biology or chemistry, a more efficient system at that opsin level?
Hardly. They are they've got everything you wish for. You can recycle them, you can use the same molecule over and over again.
What about their sensitivity?
Sensitivity is very high, and you can also make them, but actually it's pretty high in melatonin as well. If the right wavelength of light comes near it, it'll be absorbed. So that's not the major problem, I think. But one important thing is that it's very easy to make the vitamin A molecule, or retinol as it's called, be sensitive for different wavelengths. So just bend the molecules a bit here and there, and it'll shift its spectral sensitivity to wherever you want it. Other important things are, and that's not true for melatonin at all, it's kind of rigid and fixed. Other things are, they have to be not actually activated by heat, because then you get heat noise. And the vitamin A system is really good. It starts to break down when you get into long red wavelengths and near infrared, and then they start to be really heat noisy. And that's the reason why most animals don't go up there, because you mainly see your own heat, especially warm-blooded animals. They will just see a haze of their own heat if you try to see in the near infrared. Plus the world, that's another interesting thing here. The world is basically very, very bright in the near infrared. It's very dark in the ultraviolet. Everything in between, you've got colors, because certain things are bright, other things are dark. So you've got lots of contrasts in there. Up in the near infrared, everything is very pale and bright. In the ultraviolet, everything is dark and boring. Actually, the sky is bright. So the horizon is really sharp and good in the ultraviolet, which is good for some mammals. Right?
All right.
Seems with.
Oh, yeah, and I got another one too.
Yeah. Pardon? Then you always have problems, because cells can't do too many different things at the same time. So, that's a typical thing you see in evolution, that there is a division of labor. It's one cell that does two different things. Suddenly you have two cells, and one cell does one thing and the other cell does the other thing. Then you can optimize the chemistry you need, the biochemistry you need in the cell to do that particular task. And then the other cell can be optimized to do another task. So very often you have to divide these things into different cells. That's a very general, typical evolutionary mechanism, which leads to more complex things. Yeah.
Well, I already asked my question.
Okay.
I was just wondering, was when you say from intuition to what we have today, was that also when the eye started moving, like by itself, or is that later?
No, that comes with it. When you go into object vision, you really need eye movements. You need to shift your gaze. That's important. So that's one thing that you need to add. Not absolutely need, but it becomes very, very useful to be able to do it. Yeah, yeah. For many reasons. It's not good to have a fixed eye, because when you move, you get motion blur. So if you move and you fixate at one point, and then you jump to another point, and then you jump, during those jumps you don't see anything. But for most of the time you have really good vision, because the image doesn't move. And that means you can actually have slightly longer integration times, actually much longer integration times, and you can see smaller contrast differences because of that. Right? Yeah. Okay.
Another naive question per se, but I was just wondering if there's like a size or shape correlation with like the wavelength of light it can like absorb. So like I didn't know if, because you said one like certain animals can see UV, and then like certain animals can see into the
Animals with very large eyes can rarely see in the ultraviolet. No. But animals with smaller insects that have much shorter distance between the cornea, the surface of the eye, to the photoreceptors, nearly all of them see ultraviolet.
I mean, that would make sense, because the particles get shorter, they can. Like,
yeah, the main problem is that ultraviolet is absorbed in proteins. So you can't pass that ultraviolet light for very long until it's gone. before it. So there has to be something left when it reaches the photoreceptor. Otherwise, yeah, you don't see anything anyway.
All right, at this point, I think it's a good time to thank Dr. Nilsson. And please don't forget, next door, Riverview 520, Chemistry Museum. We'll have some snacks and have more opportunities to
The food is,
ask questions.
the food's in 518. The food's in 518.
We actually put it out by 520.
It's set up in the, it's right next.
So it could be that the food ended up being set at 5:15, but my students
It's
were actually
close.
bringing it to it was brought to the front of the museum. What's the
Yeah,
plan?
yeah, so they set it up.
Enjoyed that.
Thank you.
So I'm imagining,
Right,
so the
5:15,
question of is there
right next
something
to the
more
museum,
sensitive
the
or
actual
whatever?
food.
Some mechanism by which you cause a molecule to be in a state which is then, when triggered with a photon of interest, provides a cascade.
Yeah.